Scientists set out to answer a straightforward question. Take hazel eyes, sequence the DNA of people carrying them across multiple continents, compare the results, and find the common thread. It seemed like the kind of study that would close a chapter, nail down the genetic signature, confirm the inheritance pattern, write the summary. That is not what happened. What the researchers found instead was a trait that refused to behave like one, not a single dominant variant, not a clean cluster of markers. A network of interacting genes producing an outcome so variable, so resistant to prediction, that the most sophisticated genomic tools available still cannot call it correctly more than 80% of the time. For a field that can reconstruct entire population movements from ancient bone fragments, that number is striking. Hazel eyes affect roughly 5% of the global population. That alone makes them rare. But the more you examine where they appear, how they form, and what the DNA actually says, the more that rarity starts to look less like an accident and more like a consequence. A consequence of geography, of migration, of thousands of years of populations colliding at specific crossroads on the map. The global comparison did not simplify the story of hazel eyes, it deepened it. And what sits at the center of that deeper story is something science is still working to fully explain. Look directly at a hazel eye and something immediately resists categorization. It is not brown, it is not green, it is not gold. It appears to be all of them simultaneously, and then, as the light shifts, it rearranges itself into a different combination entirely. This is not a trick of perception. It is physics and biology operating at the same time, producing a result that no single label can hold. The mechanism begins with melanin. Inside a hazel iris, two forms of pigment are present and unequally distributed. Eumelanin, which produces browns and blacks, concentrates most heavily around the pupil. Pheomelanin, which produces reds and golds, spreads outward toward the iris edge. The boundary between these zones is not a clean line. It is a gradient, and that gradient is where the color complexity lives. What happens next involves light scattering. The same physical process that makes the sky appear blue, Rayleigh scattering, operates inside the iris. Light enters, strikes pigment molecules in structural tissue, and scatters at wavelengths that depend on the angle and intensity of the source. Change the lighting and the scattering shifts. The perceived color shifts with it. This is why hazel eyes change. Not because the pigment itself changes, but because the eye is not displaying a fixed color. It is generating one in real time, differently, every time the light does. The old model of eye color genetics was elegant and wrong. Brown dominant, blue recessive, two genes doing the work, inheritance predictable from a simple chart. That framework collapsed the moment researchers looked closely enough. Nowhere does it fail more completely than with hazel eyes. The primary players are OCA2 and HERC2, two genes sitting adjacent on chromosome 15 that together regulate melanin production in the iris. People with brown eyes carry variants that drive high melanin output. People with blue eyes carry variants that suppress it almost entirely. Hazel eyes land in the middle, producing intermediate levels, but OCA2 and HERC2 do not come close to telling the whole story. More than a dozen genes contribute to the final result. SLC2-4A4 and SLC4-5A2 help determine where on the color spectrum an iris ultimately lands. TYR and TYRP, one affect undertone, the difference between a warmer hazel brown and a cooler hazel green. ACIP and IRF4 influence how light scatters inside the iris itself, feeding directly into the color shifting quality that makes hazel eyes so visually unpredictable. These genes do not operate independently. They interact, suppress, and amplify each other in combinations researchers are still working to fully map. The practical result is a prediction problem. Brown and blue eyes can be called from DNA with 90 to 95% accuracy. Hazel drops that figure to between 50 and 80%. This is not a gap at the edges of the science. It sits at the center of it. Brown eyes are the original. That much is not disputed. Homo sapiens emerged with high iris melanin, a direct adaptation to equatorial UV radiation that was relentless and unforgiving. For the vast majority of human prehistory, brown was not a color choice. It was the only option available. Blue eyes are younger, but older than most people assume. A 2008 study initially placed their emergence between 6,000 and 10,000 years ago. Later research pushed that estimate back substantially. Ancient DNA research has identified blue eye-linked mutations in prehistoric European skeletal remains, predating the arrival of Neolithic farmers by thousands of years. Blue eyes were already present in Europe long before the first farmers arrived. That detail matters because of what happened when the farmers did arrive. Western hunter-gatherers, who had roamed Europe for millennia, largely carried blue-eyed ancestry. The early European farmers who migrated into the continent from Anatolia between 6,000 and 8,000 years ago largely carried brown. When these two populations met and mixed across the landscape of prehistoric Europe, something intermediate became possible for the first time. That intersection is almost certainly where hazel eyes first appeared in meaningful numbers. Not a single mutation, not a single moment, a population-level event. Blue-eyed and brown-eyed ancestry combining to produce offspring whose melanin production landed somewhere between the two extremes. The first hazel eyes were not an accident. They were the biological consequence of two worlds meeting. That collision happened across much of Europe. So why did hazel eyes concentrate so heavily in one particular corner of it? The answer is geography, and Iberia's position on the map is almost too precise to be coincidental. The peninsula sits at an exact intersection. Northern European populations moving south carried the blue-eyed genetic legacy of Western hunter-gatherers and later steppe migrants. North African populations moving north across the narrow strait separating the continents carried the brown-eyed ancestry dominant across that region. Iberia absorbed both, not once, but across multiple waves spanning thousands of years. No other landmass in Europe occupies quite that position. Climate reinforced what geography made possible. Hazel eyes are most common in temperate zones, where intermediate melanin levels carry a practical advantage. Too far north and selection pressure favors lighter eyes optimized for low-light environments. Too far south and higher melanin becomes essential protection against intense UV radiation. Iberia sits in neither extreme. It occupies a middle band where intermediate pigmentation is not just genetically possible but environmentally coherent, a zone where hazel eyes are not a compromise but a fit. Then there is the Al-Andalus period. Between 711 and 1492, nearly 8 centuries of Muslim rule brought sustained and substantial gene flow from North Africa into the peninsula. This was not brief contact. It was generations of population movement concentrated in a region already carrying multiple layers of mixed ancestry. Iberia did not invent hazel eyes. It built the conditions for them to thrive. A landmark study analyzing 271 ancient Iberians traced those conditions across 8,000 years of continuous population change. What it revealed was not a region that replaced one ancestry with another. It was a region that accumulated layers and never fully shed any of them. The sequence begins around 5,500 BCE, when the first farmers arrived from Anatolia, carrying brown-eyed ancestry and layering it over the lighter-eyed hunter-gatherer populations already present. That combination alone created expanded conditions for intermediate eye color, but what followed was more dramatic. Between 2,200 and 900 BCE, the Bronze Age brought the most significant genetic shift Iberia had ever absorbed. Incoming groups carrying steppe ancestry from the Pontic-Caspian region replaced approximately 40% of the existing Iberian gene pool. Y-DNA haplogroups that had dominated the peninsula for millennia were almost entirely displaced. The evidence suggests this migration was male-biased, accelerating paternal lineage replacement while leaving maternal ancestry more intact and more mixed. The Roman period shifted Iberian genetics eastward, increasing Mediterranean ancestry through sustained trade and mobility. North African ancestry entered more substantially during this time, partly through Roman networks, partly through earlier Phoenician and Punic presence along the coastline. Then came Al-Andalus. Nearly 8 centuries of concentrated North African gene flow created the deepest admixture layer the peninsula had yet accumulated. Each wave left something behind. Iberia folded it in, rather than erasing it. Those layers are still readable today in the DNA of modern Iberians, and visible, in some cases, in the color of their eyes. The haplogroup data makes the layering concrete. It converts migration history into measurable biological signal, and what it shows is not one ancestry or two, but multiple streams converging on a single peninsula across thousands of years. On the paternal side, R1b dominates. Between 60 and 70% of Iberian men carry this haplogroup, with the subclade R1b DF27 appearing at especially high frequencies across Spain and Portugal. This is the direct echo of that Bronze Age steppe migration, the wave that reshaped Iberian Y DNA so completely that earlier haplogroups were nearly erased. Steppe populations carried lighter pigmentation ancestry, deepening the blue-eyed genetic contribution already present from Western hunter-gatherers. The North African signal is still there. Branches of E1B1b, particularly EM81 and EV13, appear in roughly 5 to 15% of Iberian men today. Haplogroup J, both J1 and J2, accounts for another 5 to 10%, reflecting Phoenician, Punic, Roman era, and Levantine influences. These lineages carry the brown-eyed ancestry that met and mixed with lighter-eyed populations already established across the peninsula. The maternal picture reinforces everything. Haplogroup H dominates at 40 to 50%, with H1 and H3 especially prominent, reflecting deep European, Mesolithic, and Neolithic roots. U5 connects back to ancient hunter-gatherer ancestry. U6 traces directly to North African Berber populations. Both sit inside the same modern Iberian gene pool. Before I show you the last part, if you want to look at your own results and see where Hazel I ancestry fits into your bloodline, I put a full 3-volume PDF bundle over at the OriginDNA website. It shows you how to read your raw data and find matches beyond what Ancestry or 23andMe tell you. Link is in the description. But stay right here, because what comes next is the part most people completely miss. The haplogroup picture is detailed, the migration history is documented, the genes involved have been identified and named, and yet, when researchers sit down with a DNA sample and try to predict whether the person carrying it has hazel eyes, they are wrong between 20 and 50% of the time. That gap deserves more attention than it typically receives. Brown and blue eyes differ primarily at a single, well-understood genetic locus. The prediction models for those traits work because the signal is strong and the variables are limited. Hazel eyes involve a network of interacting genes whose combined behavior resists clean modeling. The best current tools call it correctly between 50 and 80% of the time. For a field that can reconstruct ancient population movements from fragmentary bone, that number is not a minor footnote. It is a fundamental limitation sitting at the center of the trait. What the prediction problem reflects is something real about the biology. Hazel eyes are not a simplified version of brown or blue with one variable swapped out. They are a genuinely different kind of genetic outcome, one produced by a system of interacting factors that does not reduce neatly to any single dominant signal. The complexity is not a gap waiting to be filled by the next study. It is a structural feature of how the trait works. Scientists expected the global DNA comparison to narrow that gap. Instead, it confirmed how wide it actually is. The more populations examined, the more combinations emerged. The prediction problem did not shrink. It scaled. Science now has the vocabulary to describe what ancient cultures could only observe. But the observation came first, and it was not wrong. Across traditions separated by geography and centuries, hazel eyes were consistently marked as different, and the qualities attached to them were surprisingly consistent. Celtic mythology associated the hazel tree with wisdom and hidden knowledge. The nuts of the hazel were said to contain all understanding of the world. People who carried hazel eyes were believed to share that quality, perceived as capable of reading what others could not see. The association was not arbitrary. Hazel eyes in Celtic populations occupied overlapping territory across Western Europe for long enough that the connection became cultural fact. Roman writers took a different angle. They described hazel eyes as containing contradictions, fierce and tender simultaneously, passionate but also contemplative. The color's refusal to settle on a single tone became a metaphor for a personality that could not be reduced to a simple read. Roman poets returned to this quality repeatedly, treating the eye's visual complexity as a signal of interior depth. Arabic poetry gave it a different name entirely: honeyed eyes. The metaphor is precise. Like honey, hazel eyes shift in color depending on how the light catches them. Warm in one moment, darker in the next. Never fully transparent, never fully opaque. What all three traditions were responding to was the same physical phenomenon, the color-changing quality that physics now explains. Ancient cultures did not have the mechanism. They had the observation, and they treated it as significant. They were not wrong to do so. If hazel eyes were rare enough to be striking, and striking enough to be attractive, basic evolutionary logic suggests they should have spread. Rare traits that confer reproductive advantage tend to become less rare over time. Hazel eyes have remained at roughly 5% of the global population for as long as the historical and genetic record allows us to measure. That stability is not an accident, it is a mechanism. The mechanism is called frequency-dependent selection. It operates on the principle that the value of a trait is partly determined by how common it is. In a population where brown eyes dominate, a hazel-eyed individual stands out. That visibility carries a reproductive advantage. More attention, more selection, more offspring carrying the relevant gene variants. But as hazel eyes become more common, they lose the rarity that generated the advantage in the first place. The selection pressure weakens. The frequency stabilizes. This produces an equilibrium. Hazel eyes remain uncommon enough to retain their signal value but do not disappear, because when they dip below a certain threshold, the advantage returns and pulls the frequency back up. Evolution is not pushing hazel eyes toward dominance or toward extinction. It is actively maintaining them at a specific range. Geography compounds this. The temperate climate zones where intermediate melanin is environmentally coherent are themselves geographically constrained. Hazel eyes cluster where the conditions support them, and those conditions do not extend uniformly across the planet. The rarity is structural. It is written into both the biology and the map. Every hazel-eyed person alive today carries something that cannot be seen directly but can be read. A biological record of populations that met, mixed, and moved across thousands of years of human history, compressed into the pigment distribution of a single iris. The hunter-gatherers who roamed Europe for millennia did not know their blue-eyed descendants would one day collide with brown-eyed farmers walking out of Anatolia. The steppe migrants who reshaped an entire peninsula's Y DNA in a single Bronze Age wave were not aware they were contributing to an eye color that would still be measurable thousands of years later. The North African populations who crossed into Iberia and remained for 8 centuries were not thinking about melanin gradients. They were living, moving, surviving, and reproducing. But the genetic consequences of all that movement are visible today in the eyes of roughly 1 person in 20 on Earth. That visibility is what makes hazel eyes unusual even among unusual traits. Most of what ancient migrations left behind requires laboratory equipment to detect. Hazel eyes do not. The record is on the surface, shifting in the light, readable by anyone paying close enough attention. The global DNA comparison confirmed what the migration history already suggested. Hazel eyes are not a single trait with a single origin. They are a convergence point. Every population that contributed ancestry to the regions where hazel eyes concentrate left a signal, and those signals interact in ways that still exceed the predictive capacity of modern genomics. The archive is real. It is also still being decoded. The science of hazel eyes did not get simpler when researchers compared DNA across the world. It got more layered, more precise in some places, and more openly unresolved in others. That is not a failure of the research. It is an honest reflection of what the trait actually is. What the global comparison confirmed is that hazel eyes are not a regional quirk or a minor pigmentation variant. They are a convergence point for some of the most significant population movements in human prehistory. Western hunter-gatherers carrying blue-eyed ancestry meeting Neolithic farmers carrying brown. Steppe migrants reshaping the genetic landscape of an entire peninsula in a single Bronze Age wave. North African populations crossing into Iberia across 8 centuries of sustained contact. Each encounter created conditions where intermediate melanin became possible, and those conditions accumulated most heavily in the places where the most layers met. The prediction problem remains. Between 50 and 80% accuracy is where the science currently stands, and that gap sits at the center of the trait, not at its edges. Ancient cultures noticed something genuinely unresolved here. Modern genomics has confirmed it with considerably more precision, but has not closed it. What hazel eyes ultimately represent is a record that has not finished being written. The history that produced them spans thousands of years. The science working to fully explain them is still catching up. That distance between what the past reveals and what the present cannot yet predict is exactly what makes them worth looking at.
Thanks for watching. Before you head out, don't let your DNA profile just sit there collecting digital dust. Tap the link in the description to download your ebook and finally make sense of it all tonight.